Hello, my name is Jason Adrians. Today is 4/8/25, and this is my DNA extraction lab, the videotape assignment. Throughout this video, I'm going to be re-simulating the lab. I didn't record the actual time that I did it, but I have a final product picture of the actual lab that I did to show the DNA and the results that I got. So to answer the questions at the very beginning, DNA is something that's found in all living organisms, and it helps basically create everything that the living organism makes. So it's the template for RNA, and then RNA creates the proteins, and that's what all the living organisms can make. Throughout this process, I added salt to the solution to help neutralize the phosphate backbone of the DNA, and that's eventually what helped it extract itself. I added detergent to break down the two membranes, the cell membrane and the nuclear membrane, which actually helped the DNA escape the cell itself. And then I added the papain powder in order to kind of purify the DNA. That helps get rid of any proteins that might be on the DNA itself. And then I added chilled alcohol to help. It doesn't mix well with water, or it doesn't mix with water, so it layers. The alcohol sits on top of the solution that I created, and then the DNA itself is insoluble in the alcohol, so then it actually helps extract the DNA and helps visualize it. I'll include the picture that I took of my final product right here after this little clip, but DNA kind of looks like a mucousy, white, stringy substance, and that you can clearly see that in the picture that I have, and I'll include it right now. So for this experiment, I used the blender method. So I took my blender cup and I added just a bit of salt. Yep, just a bit of salt. And for this, I'll simulate the dried peas with green food coloring, just to add a bit. And then I took a cup of water and added the cup of water to this blender. Then I blended it. After it was done blending, I took the cheesecloth that was provided in the lab kit. I put it over a glass like this, and then I strained the blended mixture into the glass. After straining this into the glass, then I put the detergent in. Obviously it's empty, but I put the detergent in, and then I gently mix this, just like that, or with the stirring rod, and then I put this mixture into the fridge for 10 minutes to chill. And at the same time, I had my alcohol in the freezer already chilling. So after 10 minutes, I took my mixture out of the fridge. I filled the test tube up about 1/3 of the way with the solution, and then I took my meat tenderizer, the assay that here, and filled that. I took about half the meat tenderizer and put it in the test tube. And then I took the alcohol out of the freezer. I put the alcohol into the mixture. And then after putting about the same amount that was given, so the test tube was filled up about 2/3 of the way. And then the alcohol separated out from the mixture, and then it took the DNA up to the top. Again, I have that picture, I already included it, but then it gave this white, you could see the DNA, so you could see the white stringy mucousy substance, which was the DNA, and the alcohol was separated on top of the water, so you could see everything clearly. Thanks for watching.